Hello students, welcome to EPG Pathshala. I am Dr. Nidhi Didwania from Manav Rachna International University. Today we are going to discuss about the module Ion Exchange Chromatography IEC applications from paper chromatographic techniques. So students, let us see what we are going to learn in this module. To understand the scope and types of analysis of ion exchange chromatography, to explain applications of ion exchange chromatography in environmental analysis, industrial, food flavors, pharmaceuticals, bioactive natural products, clinical analysis, cosmetics, scientific research, and to learn about advantages and disadvantages of ion exchange chromatography. In ion exchange chromatography, ionizable molecules are separated on the basis of their total charge. The similar types of molecules that would be difficult to separate by other techniques due to charge carried by the molecule of interest can be easily separated by changing buffer, pH, etc. Ion exchange chromatography process is a vital analytical technique for the discriminant and segregation of ionic compounds in alliance with ion interaction or partition and ion exclusion chromatography. It employs on charged molecules, for example, proteins, amino acids, and nucleotides and related biopolymers of different molecular sizes and nature. Ion chromatography segregation is based on electrostatic interactions between polar and ionic analytes, ionic functional groups formed to chromatographic support or gel and ions existing in the eluent. Two distinct mechanisms in the separation of ion chromatography are ion exchange due to ionic attraction and exclusion of ions due to repulsion between homogeneously charged analyte ions and the ions secured on the chromatographic reinforced execute a role in the separation in ion chromatography. Ion exchange has been the prevalent mode of ion chromatography. The separation principle is based on the development of ionic bonds between the ion exchange support sustaining the opposite charges and charged molecules. Biomolecules exhibit various degrees of interactions with charged chromatographic media owed to their variability in charge properties. Scope of ion exchange chromatography. Ion exchange chromatography envelops liquid chromatographic techniques and has been defined by the range of solutes instead of specific combinations of detection and separation modes. It is devised explicitly for the segregation of disparately charged compounds from stationary and mobile phases. The development of IEC is prompt as it provides a reliable and authentic method for the investigation of cations, anions, metal ions, amino acids, phenols, nucleotides, and alternative polar molecules. The technique has been successfully enforced to the analysis of raw materials, conflicting impurities, ions, and decandent products, analysis of cleaning solutions of production equipments, and various other applications. Immense pertinence, including average cost, tremendous performance, influential resolving ability, huge sample handling capacity, and affluent scale up conjoint automation has contrived ion exchange chromatography as one of the most significant and broadly used among all liquid chromatographic techniques. IEC is considered as mature and well-established approach for chemical, petrochemical industries, food and beverage industries, pharmaceuticals, clinical industries, and metallurgical applications. Types of analysis. Ion exchange has proven to be one of the major methods of fractionation of labile biological substances. From the introduction of the technique in the 1960s to the development of modern high-performance media, ion exchange chromatography has played a major role in the separation and purification of biomolecules and contributed significantly to our understanding of biological processes. IEC can be used for many types of analysis. Qualitative analysis of cations, anions, halides, etc. Quantitative analysis of cations, anions, halides, etc. Trace analysis and ultra-trace analysis of heavy metals, etc. Ion exchange is frequently combined with other techniques which separate according to other parameters such as size, for example, gel filtration, hydrophobicity, for example, hydrophobic interaction chromatography, or RPC, or biological activity, for example, affinity chromatography. You can see a chromatogram. Chromatogram is depicting the four main stages of ion exchange chromatography, that is equilibration, application of sample, elution, and regeneration. Protein A in the chromatogram carries the same charge as the stationary phase and does not bind. Protein B binds. Applications. Environmental. It plays a major role in softening the hard water due to the calcium, magnesium, and other divalent ions. The water is passed through cation exchanger charged with sodium ions. Calcium and magnesium ions retained in the column while sodium is exchanged. It involves complete demineralization of water by removing both cations and anions. The steps consist of passing water through acidic cation exchanger. Calcium, sodium, and magnesium ions are exchanged by hydrogen ions, and in another step, the water is passed through basic anion exchanger. Chloride, nitrite, sulfate is exchanged by hydroxide ions. Thus, IEC is used for softening hardness of water, demineralization of water, removal of interfering radicals. Significant examples. One of the significant examples of this technique is the evaluation of absolute cyanide by IEC. The cyanide is complicit to metals such as iron, and substantial digestion of acid in the existence of magnesium chloride catalyst is the requisite to liberate cyanide. The hydrocyanic acid thus resulted to evacuated by reflex distillation from the digestion matrix and absorbed by sodium hydroxide solution, which can be investigated for cyanide utilizing ion exchange chromatography with ampere metric detection. Similar technique can be utilized for fluoride and sulfide in solid samples. Acid digestion is best for the preparation of samples which are to be evaluated for cations utilizing IEC technique. Absolute nitrogen as the ammonium ion, for instance, has been investigated in environmental samples using IEC by direct evaluation of general digest matrix. Subsequently, rare earth metals and earth metals are evaluated utilizing post-column reaction. Analysis of acid rain and rainwater. The analysis of acid rain and rainwater is important application of ion exchange chromatography. The conviction of ionic constituents in rainwater is periodically used to evaluate the effects of acidification on urban and natural environments caused due to acid rain. Several researchers have developed that enables the determination of both cations and anions in atmospheric aerosols and rainwater. Analysis of terrestrial waters and ice cores from intact environments of Antarctica to measure levels of pollutants. Ion exchange chromatography has been used to analyze terrestrial waters and ice cores from intact environments of Antarctica to measure levels of pollutants. The estimation of inorganic solutes, metal species from surface water bodies, and ability to quantify different oxidation states, for example, iron, stable complexes of metals, clarifies the potentiality of the IEC technique. Determination of inorganic ions in soil. The determination of inorganic ions in soil is the vital ability to analyze the toxicity of metal ions in soil, solid waste, and sludge. The potentiality of IEC to quantify various stable metal complexes and metal oxidation states in soil extracts is benign in determination of the toxicity, fate, and transport of metals in soil. Determination of phosphorus, sulfur, nitrogen, and their analogous oxide anions, for example, phosphate sulfate and nitrate, emphasizes in soil fertility and its conditioning. Industrial. The industries has resulted in the toxicity of water, air, and soil, thus leading to increase of environmental pollution and poor quality of human life. In contemporary years, the priority of auditing and regulating environmental toxins has become prime importance in worldwide. IEC is accurate analytical technique for solving many disastrous problems like production of semiconductors, determination of heavy metals in cement, determination of petrochemicals, for example, amines, mineral acids, cyanides, etc., analysis of metallurgical components, for example, steel and alloys. Ion exchange chromatography provides the sensitive and authentic analytical procedure and techniques for the evaluation on minimum concentration levels. IEC is the beneficial instrumental evaluation technique which provides reliable methodology minimizing the detection limits. IEC is the most determined approach for the evaluation of organic and inorganic ions. Some of the significant examples of the ion exchange chromatography in industrial aspects are the evaluation of coolant ionic constituents, for example, sodium hydroxides, ammonium ion, nitrite ion, calcium ion, sulfate ion, etc. Fat acid investigation and trace sulfate evaluations in brines. IEC is broadly accepted as the specialized reference method not only for wastewater analysis or for water but has the potentiality to meet the requisite of the standards. It is nowadays increasingly recognizable as in the laboratories of industries like evaluation of heavy metals and cyanide polyphosphates in electrochemical and tannery waste, chemical purity and heavy metal analysis in fertilizers, analysis of alkaline earth metals, sulfur and chlorine in paper industry. Food flavors. Ion exchange chromatography is relevant technique in food and nutrition industry as compared to other techniques in terms of accuracy, sensitivity, and speed. IEC is beneficial when the analytes group to be investigated within the same chromatographic run needs slight or non-sample treatment. Food, beverages, raw materials are chemically analyzed for distinct variety of constituents. The presence of contaminating constituents, for example, heavy metals, antibiotics, adulterants, and antibiotics are best analyzed by IEC. The major application areas are determination of heavy metal limitations in dairy products. Evaluation of sulfates, chlorides, nitrates, and phosphates in sweeteners and sugar syrups. Acid content, for example, lactic, malic, citric, and acetic acid in coffees, food, and other beverages. Fluoride content in teas. Ion exchange chromatography, being the derivative of high-performance liquid chromatography, has the advantages of speed, sensitivity, exactitude, reproducibility, etc. It can be applied for the purification and separation if charged molecules, for example, enzymes, proteins, DNA, nucleotides, peptides, vitamins, antibiotics, etc. Detection of glucose, lactose, and galactose in dairy products in lactose-free category, arsenic in rice, and phosphoric acid in cold drinks, amino acid content in specialized nutritional products, presence of carbonates in drinking waters. Pharmaceuticals. Ion exchange chromatography has an essential role in pharmaceutical industries for the evaluation of integrity of medical mixing, determination of selected constituents, and in control production of drugs. IEC is a well-established process that ensures the drug has been manufactured as per sanctioned procedures and also certifies cleaning of the residues from the equipment utilized during the process of manufacturing. Some of the examples of the use of ion exchange chromatography in pharmaceuticals are: it separates the cations in water solution for injectables. It analyzes the final drug formulation and help in the pharmaceutical industry. IEC has been occupied its place in drugs in the form of capsules and tablets in order to evaluate the content of drug dissolve with time period. It also detects and quantifies the inactive ingredients utilized in medicine formulations. It has made the detection of impurities or constituents that are not chemical entity easy. It provides the insights about the contents of drugs that should be given to the patient per day. IEC is entrenched approach for the investigation of charged species, for example, vigorous acid ions, sulfate chloride, fluoride, phosphate, etc. This technique is suitable for the separation of charged particles of weak nature, for example, carboxylic acids. The uncharged species are separated on the basis of three mechanisms, for example, Donan exclusion, adsorption, and steric exclusion or segregation process. The charged species are passed through the column unrefined. Manufacturing process of drugs utilizes various excipients, for example, citrate buffers or phosphates in inconjugatables, filters, for example, calcium phosphates and calcium sulfates. Ion exchange chromatography process provides a simple method for these manufacturing processes of drugs. Applications in medical. Anion exchange chromatography is used in treatment of ulcers. Cation exchange chromatography is used for removing sodium ions and treating hypertension and edema. Bioactive natural products. These products are the chief source of functional foods, food additives, and new types of drugs. They consist of secondary metabolites, for example, alkaloids, sterols, flavonoids, sterols, etc., and animals and plants generated through biological processes. IEC is one of the most often utilized techniques for the separation of natural products in pure form. It is the core fundamental and efficacious technique as it is inexpensive and offers high recoveries of key analytes. The applications of IEC are as stated under. The cation exchange column is utilized to segregate thiamine from riboflavin. IEC is broadly used for the separation of biological large molecules like proteins, polysaccharides and proteins, etc. It has the potentiality to extract and isolate mycotoxins produced by the fungus, for example, Aspergillus niger. Recovery of basic and acidic compounds from plant extracts utilizing solid phase reagents. The classic application is the extraction of noxious indolizidine alkaloid, swanisonine from plant constituents, etc. Ion exchange chromatography is used for the extensive extraction of natural products from limonene, glucosides and agricultural source. It results in valuable recovery yield from citrus molasses. Clinical analysis. Ion exchange chromatography provides well-established applications in clinical perspectives. It provides a huge range for the prospects of selection in mobile and stationary phases. It aids in solving distinct separation problems when combined with various detection techniques. It can be utilized to evaluate various ions and substances in the pharmaceuticals and clinical aspects. It contributes the availability of great extent stationary phases and precise detectors. IEC has marked its presence in clinical aspects due to various reasons: elementary sample preparation, reduced sample volumes, prevention of hazardous chemicals, flexibility in utilizing reaction options, so on, variation of sample metrics to be investigated. IEC provides aid in determining various organic and inorganic constituents in clinical samples. It provides the maximal rates of diagnosis and permits the diagnosis of amino acidopathies as compared to other techniques with thin layer chromatography declined to detect as the concentration of few amino acids was very little. IEC has considerably improved diagnostic fate in clinical perspectives to a large extent. It is used as diagnostic aid in gastric acidity test. cosmetics. The accuracy, subtlety, and suitability for the investigation of organic acids have remarked the existence of ion exchange chromatography in cosmetic industry. The detection of organic acids, for example, formic acid, lactic acid, malic acid, citric acid, glycolic acid, tartaric acid, etc. in cosmetic samples by ion exchange chromatography methodology has abolished conductivity detection. One of the significant example is the determination of thioglycolic acid in cosmetic samples by IEC is remarkable in suppressing conductivity detection. Ion exchange chromatography benefits whenever the analytical problem consists of elimination or isolation of an ionic compound. Ion exchange can be used for the selective desorption of individual anions or cations in the mixtures of ion. One of the leading features of this technology is the constituents of shampoo are deserted in an unaltered chemical form. IEC has well established its utilization in herbal and Ayurvedic cosmetics. IEC has been used for the segregation of distinct ionic or the mixture of non-ionic and ionic compounds such as surface active agents. It is highly sensitive technique for the detection and evaluation of nitrosamines in our environment. NDELA in cosmetics is the impurity in samples of cosmetics. It is adsorbed by storing basic anion exchange resin. IEC facilitates the cleanup process of cosmetics by recovery of various other compounds utilized in formulations of cosmetics. Scientific research. Ion exchange chromatography has established the leading role in scientific research area. For instance, the purified form of immunoglobulin G is efficaciously utilized in passive immunization process. It provides to be effective in using DEAE Cefadex A50 to yield pure immunoglobulin G. With the new advancements in technology, a quantitative research of evolution of amino acids in humans is made easy by ion exchange chromatography technique. Investigation of amino acids in the urine and plasma is one of the remarkable features of IEC. Determination of amino acids in physiologic fluids by IEC. IEC also facilitate a lot in the scientific area, not only in medical but also in various other fields. Advantages of ion exchange chromatography. This approach has been utilized for the evaluation of anions and cations, implicating metal ions, hydroxyl compounds, peptides, organic acids, nucleotides, and alcohols, etc. It has been auspiciously applied to the investigation of deteriorated products, raw materials, impurities, bulk active ingredients, etc. at various stages of production steps, in addition with the evaluation of the cleaning solutions of production apparatus, desolated streams, container compatibility, including other utilizations. Ion exchange chromatography has become one of the chief chromatography as compared to other liquid chromatography approaches because of average cost, huge sample handling capability, easy automation as well as scale-up, dynamic resolving ability, extensive applicability in addition with high throughput application scheme and broad applicability, It can be used at all the phases and proportions of purification. It is a non-denaturing process. It can be maintained by altering the salt concentration, pH, and the ion exchange media. It provides high selectivity and can resolve molecules with little variations in charge. It can serve as intensifying step. A huge volume of diluted sample can be enforced to a media and the adsorbed proteins afterwards eluted in petite volume. Eco-friendly technique as it deals with substances existing in water like longer life of resin and economical maintenance. Disadvantages of IEC. Despite of broad use of IEC, the mechanism of segregation has not completely been enlightened, and extensive effort has been made to depict the ion exchange process more theoretically. One of the major disadvantages of IEC technique is that it presents no direct information on the situation occurring at the stationary phase surface, as the ion exchange symmetry is always resolute by the balance between the eluent and solute interaction with the active site resins. One of the main disadvantages of ion exchange chromatography is that ion exchange columns must be loaded in low salt buffers because binding to ion exchange resins depends on electrostatic interactions between the stationary phase and proteins of interest. For some applications, this restriction may require a buffer exchange step prior to IEC. Weak ion exchangers highly depend on pH, and these resins rapidly lose capacity and resolution when working outside of their optimal pH range, which is a limitation of ion exchange chromatography. Other major disadvantages are properties and nature of ion exchange resins. Chemicals and equipments are expensive. Turbidity should be maintained below 10 parts per million. Nature of ions exchanged. Substances, for example, organic matter or resins, can filthy effect to resin. So students, let us summarize what we have learnt in this module. We can see that there is an unlimited scope for ion exchange chromatography. It is very helpful in both qualitative, quantitative, as well as trace and ultra-trace analysis of the compounds. There are a wide variety of applications of IEC in different fields like pharmaceuticals, food, flavors, fragrances, environmental analysis, forensic science, etc. Ion exchange chromatography has numerous advantages and a few of disadvantages. Thanks.